All right, I just wanted to stick in Unit 4A where we talk about serology and immunochemistry methods. So let's go ahead and do that now. This is Unit 4A. So let's go ahead and start with a couple terms. Agglutination is antibodies clumped with antigens that are naturally occurring on an organism. That's called the direct method. Or they clump with antigens that are artificially placed on latex or charcoal particles. That's called an indirect or passive method. Then we have hemagglutination, which has to do with cells. Hemagglutination detects antibodies to antigens naturally occurring on red blood cells. That's called direct hemagglutination, like the heterophile test for infectious mononucleosis, or an indirect or passive test where antibodies react with antigens on blood cells that have been artificially placed on the cell. You'll notice that agglutination has to do with a particulate in nature, and hemagglutination has to do with an antigen that's cellular in nature. Okay, so we have direct agglutination, indirect agglutination, direct hemagglutination, and indirect hemagglutination. Agglutination happens in two steps. The first step is called sensitization, and the second step is called lattice formation. In the first step, we have an antibody that physically binds to a cell or particulate, thus sensitizing it. It's the physical attachment of antigen and antibody. In lattice formation, for this to happen, there has to be an appropriate antigen-antibody concentration. The antibody size is also a consideration, and IgM, because it's a pentamer, binds better because it's larger. The number of combining sites on an antibody and the number of combining sites on an antigen or cell or particulate would have to be equal. Okay, tiny particles from chemical bonds begin to form much larger bound particles until it has the potential to become one solid particle through cross-linking. When patient's serum contains antibody and will bind to antigen, red blood cells from another source are added, and since antibodies are bound, they will not lyse other RBCs. This is the principle for hemolysis and neutralization. When a red cell is used as an indicator system to show that an antigen-antibody reaction has occurred. Here, patient serum containing antibody, when added to a specific antigen, will bind. Red cells from a non-patient source are added to serial dilutions of patient samples. Red cells at the endpoint that do not lyse are neutralized by the antibody and bound up and will not succumb to lysing. This methodology is called hemolysis and naturalization. There's a couple type of labeling techniques that I'm going to address here. One is called immunofluorescence and direct immunofluorescent assay. That's also known as the DFA. And then we have immunofluorescence and indirect immunofluorescent assay, which is the IFA. In the DFA, we have antigen-antibody reactions that are seen microscopically with the conjugated antibody. The fluorochrome is fluorescein isothiocyanate, also known as FITC. The patient directly collects the samples and provides the antigen source. Some of those antigen sources could be nasopharyngeal cells like the columnar epithelial cells for RSV, genital cells or endocervical cells for the visualization of chlamydia, and those are just a few examples. The antibody in the kit is tagged with fluorescein isothiocyanate, and if the patient is positive, the specimen is going to fluoresce. Some tests that we can use for DFA, or some bacterial infections that we can test for using DFA, would be RSV, which is a virus, herpes, chlamydia, and CMV, which is a virus. IFA, on the other hand, is when we have antibodies in patient serum that link up to a fluorochrome in a donor cell or parts of a donor cell that is rich in the antigen. For example, if you have a person with lupus that makes anti-double-stranded DNA antibody, this patient's serum is going to fluoresce with the rich and plentiful source of double-stranded DNA found in the nucleus of the donor Cryptidia protozoan cells. Cells are donor cells like human epithelial carcinoma cells or Cryptidia protozoan cells. ELISA stands for Enzyme-Linked Immunosorbent Assay, and it's also known as EIA. It's designed to detect antigens or antibodies by producing an enzyme-triggered color change. It uses technology that provides an objective measurement of light where we're looking for optical density and provides a numerical result. It's generally used to measure total antibody and body fluids. Remember, if IgM rises, it's an acute disease, and if IgM rises, it's during the convalescent stage of the disease. Here we can evaluate the catalytic properties of enzymes to detect and quantitate immunologic reactions. This enzyme-labeled antibody or enzyme-labeled conjugate is used. The enzyme with its substrate detects the presence and quantity of antigen or antibody in patient's serum. We have three types of ELISA. One is called non-competitive, one is called competitive, and one is called capture ELISA. Capture is sometimes also known as sandwich ELISA. In the sandwich ELISA, we are looking for antigen of a pathogen. The antibody lines the wells, the substrate is added, unbound antibody is washed away, and the conjugate enhances the color reaction. One type of immunodiffusion method is called RID, radial immunodiffusion, and it's used to quantify antibodies or other things made out of proteins. Here we have a protein substance such as antigen that is placed inside a gel media with a corresponding antibody. As the antigen starts to diffuse from the well, the antigen-antibody combining starts to occur, where you start to see precipitate. When equivalence between antigen and antibody binding has occurred, you will see a precipitation ring that can be observed. That ring can be measured and compared to a package insert, and we can actually tell how much of that protein is present in the patient's serum based on the diameter of that precipitate. Now, RID can be used to measure all kinds of proteins, including antibody levels, complement, CRP, and other substances made out of proteins.